I'm Giselle, and I'm going to teach you about recombinant protein. So what is a recombinant protein? This protein is expressed by genetically engineered DNA. What is the purpose of recombinant proteins? The proteins are not naturally produced in the amounts used for purification. So how do we conquer this challenge? We force the cell to synthesize them. Good hosts for these genetically engineered DNAs. Some good hosts include bacteria, yeast, animal cells, and insect cells, specifically E. coli and SF9 cells.
E. coli is the first option because it is easy to grow and easy to modify. For insect cells, it can support eukaryotic cells, however, expression is less consistent.
E. coli again is the first option because they are perfect for the production of small proteins that do not require post-transitional modification. But given that they do not post-transitionally modify proteins, they will not be suitable for the synthesis of eukaryotic proteins that require disulfide bonds, phosphorylation, glycosylation, protein chaperones, or proper folding. Insect cells, meanwhile, can perform a variety of post-transitional modifications, thus SF9 cells can support production of properly folded and active recombinant eukaryotic proteins. This would be the best host for mammalian protein expression.
So from previous talk, it is now obvious that E. coli is the winner. Now that we have chosen a host, we can start to modify the cell. This is done through a process called transformation. Transformation occurs when the bacterial cell E. coli ingests foreign DNA from its surroundings. Keep in mind that transformation can occur in certain types of bacteria. We will be talking about the transformation in a controlled environment like a laboratory. In the lab, the E. coli and the plasmid are added into a tube. The tube will then be put through a transition of cold to hot to cold. It is important to make the transition from ice to water bath to ice very fast in order to create the heat shock. The heat shock will then cause pores to open up from the cell membrane, of course, to open up and allow the insertion of plasmids to come in and complete transformation.
What is a plasmid? A plasmid is a circular piece of DNA that includes these major components: an origin of replication, a resistance gene, which usually codes for an antibiotic resistant like ampicillin, and the multiple cloning site, or MSC. This is your recombinant DNA. In order to express these genes, we need expression vectors, which are the lac promoter. This allows us to control the timing of the recombinant protein expression, and the protein tag. This tag helps us scientists detect the expression of recombinant proteins. Now it is time to express the proteins. In order to express these proteins, an initial culture is grown from a single bacterial transformed colony. This is then used to initiate a larger culture that is grown to mid-log phase. This phase is where the recombinant protein is induced by the addition of IPTG in the medium. In order to determine what phase the bacterial growth is in, we use a spectrophotometer to measure absorbance. The growth phase is the point the cells are dividing rapidly and production of the protein will be optimal. This is also known as the mid-log phase. So, like I said before, this is done to produce large quantities of purified proteins for biomedical research. Thanks so much for listening.